Cool. All right. So she told us that we're being recorded now. So now I'm going to hop in to a screen sharing mode. Can everyone see my screen? Okay. Cool. So feel free to interrupt me and stop me whenever. I'm essentially just going to go through the lecture, just like we would during any other lab. And we can talk a little bit about the actual worksheet as well. So I'm going to hop into this mode. Oh, my screen went black. I can't see anything. Can you all see? Do you see this? Do you see the screen, like the PowerPoint, or do you see the presentation mode?
Presentation mode.
Oh, weird. How about now?
Yeah, it's out of presentation mode. I think we can see all the slides at the bottom, or at least I can.
Okay, so you like see it as in like if I was giving a presentation, you'd see it on the screen.
Oh no, it was like that before.
Yeah, it's like if you're editing it, it shows all the slides at once. It's like that type of mode.
Yeah, I think you're in presentation.
Yeah, it's this.
Yeah, now it's back. Now it's back.
So now this is good.
Yes. Sorry about that.
Sorry about that. Okay, cool. So yeah, so today we're gonna sort of go through the gymnosperms. And again, just to reorient everyone, the gymnosperms are, if we only look at extant species, they are a monophyletic group and they're sister to the flowering plants. But remember, if we look at the a lot of the extinct groups, they sort of branch off. This is not a really good description of this, but a lot of the extinct groups will branch off sort of here, in between the angiosperms and the conifers. So that renders, once we take the extinct taxa into account, it renders the gymnosperms non-monophyletic. So that's an important distinction. And again, just like I'm going back to this phylogeny slide, but you just sort of to walk through everything, right? We have algae, sister group to the bryophytes, right? Remember mosses, hornworts, liverworts, sister group to what are termed the seedless vascular plants, which are, again, actually the lycophytes are supposed to be here, and then sister to the ferns, and then we have the angiosperms and the gymnosperms diverging right here. And this is again around 150, 300 million years ago, essentially, right here. And the gymnosperms are essentially divided into 4 groups. So you have the cycads, which we'll get into those, the ginkgos, the conifers, and the netales. And the netales are like these super weird plants that I'll kind of get into, and we talked about it a little bit in lab, in lecture, but they're just like these weirdos that used to fall out as people used to think they were sister group to the angiosperms, again because they have some of them have vessels, reticulate veined leaves. They have some sort of this double fertilization, and they have some other things like fleshy seed coats that look like fruits. And then you have like sort of the most common, what we think of gymnosperms are the conifers. Then we have the sole ginkgo, which is just one species, Ginkgo biloba. And again, the cycads, which used to be very, very diverse, but are now actually relatively not as species rich. So the one thing with cycads is that they all, for the most part, have, actually, I think every single species has compound leaves. I don't think they vary in how compound they are. Some are more dissected than others, but they're all pinnately compound, as you can see here. And there is a good bit of diversity, but you can sort of see they have the same gestalt, right? And again, they used to be very abundant in the Jurassic. So if you see any illustration, artistic rendition of a landscape in the Jurassic, you'll see all these dinosaurs and and you'll see some vegetation, but you'll also see a lot of cycads and a lot of cycadioid-like things that look like this. But that's just to drive the point home that these were, again, they had their heyday, about 130 million years ago or so.
Jacob, I had a quick question about leaves.
Yeah.
Sorry if this derails anything.
No, it's fine.
There was one question on the quiz about broad leaves. I was just wondering what specifically differentiates broadleaves from like needles, or just kind of what's the breakdown there?
So when we think of broadleaves, we generally think of things that are large and multi-veined. And when we think of needles, we generally think of leaves that are sort of highly modified into being like needles, right? Like just small, elongate, generally few-veined, rigid structures. So it's more of a qualitative thing than a quantitative thing. But these would sort of be considered broad leaves.
Oh, okay.
Yeah, compared to the other gymnosperms. And for instance, ginkgo would be, I think, let's see. Ginkgo would be, you can sort of see the leaves down here, right? They would be broad leaves. And that's sort of compared to, just going to fly through this for a second, compared to something like this, right? Which are, this is sort of a needle. It's not a broadleaf. Again, it's a good term, I think, because you can easily, easily distinguish between something with a broadleaf and something with a needle. But for the most part, it's relatively qualitative.
Okay, thank you.
Yeah. Okay, so the way that pollination occurs in cycads is pretty interesting. So they have all of these. So generally, they're male cones. Or they're rather what we like to term their pollen cones are going to be these very tight, compact structures that, again, they'll open and close based on like temperature. And this allows these generally weevils of sorts to, here they drew like beetles, but I think mostly it's like weevil pollinated. But weevils will sort of go in and they'll get rewarded with some sort of reward, and they'll get coated with pollen, and then they'll go over to the pistillate, or not the pistillate, but the ovulate cones. And then they'll fertilize those ovules with that pollen. So generally, I think historically, cycads used to be, people used to think that cycads were wind pollinated, but careful observations and experiments found that they're actually insect pollinated, which is kind of interesting. So this, I think Minya did this. It's I guess it's one of her favorite trees. It's a fantastic tree. But I think my favorite tree is Metasequoia glyptostroboides, the dawn redwood, which we have photos of it in this lab, and it's commonly cultivated in the temperate region. But this is a fantastic tree, the Ginkgo biloba. Here are just some nice photos. They can be massive, and they do this really cool thing where, well, first of all, they have, as you can see, beautiful fall foliage. And the trees in Massachusetts do that too. They have the same color. But they do this really cool thing, and I don't think anyone really knows why they do this, but essentially, if you get a really cold night early on in the fall, all of the leaves the next day, if it's like really cool in the night and then really warm in the next day, or really sunny and warm in the next day, they'll all fall at the same time. And it's just like they rain down, and it's really, really, really cool. I got the pleasure of seeing this at the Arboretum, I think 2 years ago or something. It's just a really cool phenomenon. I don't think many people know why, but it creates this just beautiful scene, of like yellow raining leaves. So yeah, so now the Netales. So the Netales are just, I guess, they're the trolls of the plant world. and what we mean by that is they are evolutionarily most closely related to conifers in particular. There's still some question as to like, are they most closely related to conifers, or are they most closely related to the other gymnosperms, or are they most closely related to this one subset of conifers? So the evolutionary relationships in between the Nitales and the conifers and the gymnosperms are sort of in flux. But that being said, we know for sure that Nitales are they are gymnosperms. Historically, they used to be thought to be angiosperms, and again, that's because of a couple of these traits. Some of these traits include, like I said, the presence of vessel elements, right? So these really long, really wide water conducting tubes that lack pit membranes at their perforation plates. So this allows water to move more rapidly or more efficiently. And so that's one trait. The other trait is again, you can see these what would be termed broad leaves. And this is obviously doesn't look like a conifer with those leaves. And then the other things that they possess are a weird form of double fertilization. So again, we talked about double fertilization with angiosperms in creating endosperm, but my advisor, Ned Friedman, actually found that double fertilization does occur in the Nitales, but in weird ways. And then there's some other things which we'll look at too, which is like the presence of, sometimes they have bisporangiate strobili, which, remember, if we think about what a flower is just a bisporangiate strobilus, where you have sporangia that produce megaspores and sporangia that produce microspores. So some of the Nitales do that too. And specifically within the Nitales, Welwitschia is this super crazy species that grows in the Namibian desert. And it's one individual. So it has these intercalary meristems, which you can see right here. Do you see how it's like lighter green down there? So that it just produces 2 leaves, and then for the rest of its life, which could be like hundreds and hundreds, maybe even thousands of years, it just keeps pushing out. It keeps dividing paraclinally here and here to create more lamina. So that you can see the oldest portion of the leaves are down here, and the youngest are up here, and it's quite a remarkable plant. It's just kind of a bizarre plant, and like nothing else grows out here. So you just like see these, and you can see them in the back, right? You see like that's a patch of it, that's a patch of it, that's a patch over here, over there. It's kind of crazy. And I think, if you looked at it from like a satellite image, you would sort of see all these just splotches of, all these splotches of Welwitschia in the desert. And I guess they can get pretty massive. I've never seen one of these in the wild, but we have some in the greenhouse, which maybe when everything calms down next semester in the fall, some of you can go check those out. So another member of the Nitales is Ephedra. So Ephedra is really weird. So one thing to keep in mind is that all 3 members of the Nitales, Welwitschia, Ephedra, and Needham, are like couldn't be more different from each other. Like if you want to talk about like grouping these, things, it doesn't really work well. So I can't remember who figured it out, but I think like some botanist figured it out in the early 1900s that these 3 plants go together, these 3 genera go together. But I don't know how. So essentially, Ephedra is really weird. It has these extremely reduced leaves. So do you see these small? You can see it better in this picture on the left. They have these small reddish scales, and those are the leaves. And the stem is photosynthetic, right? So are you guys? I moved like the row of your faces up. Is it like blocking the PowerPoint, or is it okay? It's okay? Okay, cool. So Ephedra is really cool. Like, it has these strobili, which have these sort of outer bracts, if you will, and these outer bracts sort of swell up almost like a berry, and it sort of looks like a fruit, right? Because like, what is a fruit besides, leaf tissue, a carpel that's surrounding an ovule, and then it swells up and protects it. So this is kind of what it's doing, right? It's almost like another independent evolution of the carpel, but again, it's not completely surrounding it, and it's very it's not the same. It's just sort of a convergence, if you will. And here you can really see these pollen droplets, right? So what's going to happen is the pollen's going to land here, and then the droplet's going to get sort of sucked up into the ovule, and the egg will get fertilized by the sperm in the pollen grain. Okay, Needham. Needham's just, like if you walked, past a Needham in the forest, which they live in, I think they live in like New Caledonia and places like that. Just sort of like, they're old world tropic plants. But I mean, if you didn't know what you were looking for and you walked past this, you would say, oh, this is an angiosperm, right? They have these broad leaves. They're opposite. They're reticulate veined, right? I don't think there's a single other. Oh, that may not be true. There are not many gymnosperms that have reticulate veined leaves, but Needham does. Okay. Oh, so they also have these really interesting strobili, right? And you can sort of imagine how this would be related to a pine cone, right? If you just sort of take those internodes and collapse them, and then you maybe make those branches of those little leaves a little bit bigger and hard and brown, you can sort of create a little pine cone from something like this. But essentially what you're seeing here is, at least here on this central picture, you have these ovules right there, if you all see them, sort of pointing to them. And once they get fertilized, they swell up and they develop into these what look like berries. But again, these are individual ovules with a fleshy seed coat. And there might be a second like fleshy leaf material around it that's sort of like a carpel, but it's not a carpel. Okay, then we have what we think of gymnosperms, right? These are the conifers, right? Sort of the pine trees and things like this. So the way that this PowerPoint is organized, it sort of hops into like some of the things we're drawing and doing in lab. So I guess what we could do is, let me pop out of this for a second. Oh, don't know what happened. Okay. So I'm going to pull up the, this thing, the lab worksheet. So just like, I'm going to hop through the lab worksheet real quick, and then we can sort of continue with the PowerPoint. But essentially, there are 3 activities in the lab today, or whenever you guys want to do it. And they're broken up into different activities. So the first activity is just essentially, coming back to our theme of the phytomer. So we're going to look at some of the different images that we have in the online resources. Do you all have access to that? Have you seen? Okay, cool. So it should be broken up into 2 categories. There should be activity 1 and activity 2. So for activity 1, we essentially just want you to choose one of the taxa, one of the species folders, and go through and sort of draw some of the things you're seeing, right? So draw how the leaves or the needles are arranged. Are they on short shoots? Are they on long shoots? What's the phyllotaxy? If there are short and long shoots, what's going on with the internodes there? Are some of these shoots subtended by leaves? I mean, we know that branches are always in the axis of a leaf, right? We have a leaf and then a node, and then at the node there's a bud, and the bud will break and become an axillary branch. So generally that should be true. Yeah, so this is essentially what we want you to do, and that's what this slide is saying, right? Choose at least one of the different specimens, draw and describe the phytomers. How do the phytomers relate to each other? And then we'll get into the reproductive features later on in the lab. So I guess some of the, I'm going to pop up into this presenter mode again. Let's see if this works. Does my screen go completely black for you guys when that happens? Oh, it doesn't? Okay, it does for me. Okay, so some of the things that, you know, I want you all to look at are sort of like, what do we have going on here, right? There's a lot of things going on. We have all these different types of scars. We have these weird clusters. Are these branches? Are these you know, why are these clusters of leaves? What's going on here? So essentially, there's a couple different types of scars. So you can see right down here and right up here, these scars that seem to sort of overlap significantly. So those are the bud scale scars, right? So in conifers that are temperate, there needs to be a bud that's protecting the developed leaves for that's protecting the following year's needles or leaves. And generally, when the branches form for next year, it'll leave a whole bunch of scars that you can see, right? So here's one set, and then this was all 1 year's growth, and then there's the scars from the buds that were set for the next year, right? And then this is all the next year's growth. And then you have these other things that are these little, I guess, nubbins, and these are the fascicle scars, right? So what's a fascicle? Well, a fascicle is just a cluster of needles. And, you know, for conifers, we tend to have these names, these terms, right? We're just calling it a fascicle, but it's really just a bundle of needles. So, you know, one question is, well, what's the phyllotaxy? What's going on here? So if we know that this is a bundle of leaves, right, or a bunch of leaves, there has to be some sort of like internode, right? There has to be nodes, internodes, because we know that leaves are born on shoots, and shoots have an arrangement where we have, you know, leaf, node, internode, leaf, node, internode. So essentially what's happening is in fascicles, in pines in particular, you have this small scale leaf right here, and then you have a node right here. And from the node departs a small determinate shoot. So it's not a short shoot, because a short shoot, we'll get into that, a short shoot essentially is indeterminate. It will continue to proliferate forever. But fascicles are similar to shoots, to short shoots, but they are determinate, right? That's why, if some of you remember at the Harvard Forest, when we were trying to find, you know, how many years of needles are on this conifer, we really couldn't go back that far, because these short shoots are determinate. So they'll eventually fall off, and then generally nothing will sort of grow from this node again. And again, let me just hop out of presenter mode. I want to compare this to an actual short shoot, which we have in like the larches, okay? So if you see here, the difference between a fascicle and a short shoot is, like I said earlier, that the short shoots are indeterminate and the fascicles are determinate. So if you look here, this is a short shoot, this is a long shoot. This is a short shoot, this is a short shoot, this is a short shoot, this is a short shoot. And so essentially the needles are produced on this in sort of in whorls on this off of this short shoot, and then in the winter they'll fall off, and then the next year another set of whorls will be produced right here. So they have very condensed internodes, and they're indeterminate. And again, that's fundamentally distinct from a fascicle, which is a determinate, essentially a determinate cluster, right? So the terms, it's sort of just like a matter of scale and a matter of, is it determinate or not determinate, essentially. But you can imagine.
Long shoot again?
Sorry?
Oh, what was the long shoot?
Oh, on the larynx?
Yeah.
The long shoot was just the whole branch. So like this would be termed the long shoot, right? And then these are short shoots off of it. But you can see like right there, that was at one point in time a short shoot. And you can tell because of all of these highly condensed internodes and many, many rounds of leaf scars, right? So short shoots don't have to be short shoots their whole life. They can, like perhaps maybe in 5 years, in 20 years, in 100 years, this shoot decides to, sprout out, and this will become another long shoot. So it's not a once you're a short shoot, you're always a short shoot. It can sort of go back and forth. It can't go back and forth. It can go generally from short shoots to long shoots. You wouldn't necessarily go from a long shoot and then terminate in a short shoot. That wouldn't. You can't go back. But yeah. Any other questions? Okay. So yeah. So, and you can tell that, so the phyllotaxy here, if we just look at the scale leaves, they're spiral. So that means that the fascicles are going to also be born in a spiral fashion. Because, right, we know the phytomer structure. It's leaf, node, bud, internode. And if we know that these fascicles are buds, or branches that were derived from buds, then we know that the branch phyllotaxy is going to be the exact same as the leaf phyllotaxy. Yes, this is exactly what I just said. Okay. Okay. Question: are these fascicles or new branches? What do we think? Well, let me ask you another question. What's going on back here? What is this, a leaf? These are leaves, right? So if we know that leaves are sort of, they seem to be born on a stem, and it doesn't seem to be that there's like a short shoot, long shoot system, or that there's sort of clusters of leaves. If that's the case, then what do you think these are?
Fascicles, maybe. I'm not really sure how that helps you.
So I guess, so they're branches, but the reason why you would know that is, well, from this exact, it would be kind of hard to actually tell from this, but if we know that the leaf arrangement on all of these, if we know that leaves are born on the stems in sort of this world fashion for this plant, and we have these buds that are starting to break, that look like they're subtending a leaf, just based on sort of what's going on back here. I think it can inform our understanding of what this actually is. This is kind of not a great slide to like articulate this, but I guess so the answer would be it's a branch, and just look at the other leaves and look at like how everything else is arranged, essentially. But not a great question. Okay, so okay, I want to skip this for a second. I want to hop here. I want to talk about using dichotomous keys, right? So if we look at the lab sheet, and we look at the sort of the last portion of the activity 1, you have to identify your gymnosperm. So there should be a folder that you have access to that has a whole bunch of unidentified taxa, like taxa A, taxon B, taxon C, etc. And you essentially want to just choose a folder and go through and try to key out what species it is. And the way to use a key like this is generally termed a dichotomous key or a polycotomous key if it has more than 2 answers. And the way you sort of read something like this is like such. Let me zoom in a little bit, and then we'll go through. So you essentially have things ordered in nested like facts, right? So you read these like they are facts, and you ask, you sort of just go down the list, and you first read it. You first ask the question, or you first state the fact: needles reduced to scales. If you say, okay, yes, then this is a very simple one, right? It gets you right to here. That's thuya. But if you say no, then you move directly down to the next fact that is in the same indented line as this, right? So then you say, okay, needles not reduced to scales. That fact is incorrect for the thing I'm looking at. So I move down to here. Then I say, needles born in clusters. No. So then I go down to here. Needles born singly. Yes. Okay. So then from here, if I say yes, then I move one indent over, and then I ask this question: round needles. No. Then I go down to B, right? I don't go back here because I said yes to this fact. I stay within this fact. You never leave the facts if you have determined with 100% accuracy that that's the correct thing. So I will say that sometimes I've been keying things out, and I'm here and I'm like, oh yeah, this is 100% what it is. And I like end up in this rabbit hole of things because like I made a mistake up here. It was actually broadleaves instead of needles. And then I made a mistake, and then I'm like down this rabbit hole. So you do have to sort of, you can make a mistake later on and you'll have to fix that. But for something like this is relatively simple. But anyway, that's how you use a key. And so you start at the outermost indented region, and then you just sort of move your way in. And then finally, if we say, okay, needles born singly, yes. Rounded needles, no. Flat needles with no stalks, no. Flat needles with stalks, yes. Then we just go down to this one and we say clear white stripes underside, yes. Okay, suga. And that's how you use a dichotomous key. This is actually a really useful skill, not just for this lab, but also if you're interested in learning a new flora. Let's say you move to New Mexico and you want to learn the New Mexican flora. You can purchase a book that has all of the species, and they'll almost always be organized in this framework. So it's really important to learn how to use a dichotomous key like this. Okay, so that's that. Okay, so now let's talk about reproductive features. So as we know, the conifers and the gymnosperms they don't produce flowers, right? That's one of the things that distinguish them from angiosperms. They don't produce flowers, they don't produce carpels, and they generally, within the conifers at least, they produce their reproductive parts on cones. And a cone, as we know, is just a strobilus. And as we know from the lycophytes, we know that a strobilus is just an axis like this with a small subtending leaf and a sporangium, right? That's exactly what we saw in the lycophytes, just like this one on the left. This is Selaginella, and if you remember from our lycophyte lab, if you cut longitudinally through one of those sporophores, one of those strobili, you would see that each sporangium is subtended by a leaf. And that's no different from what's going on in this simple strobolus. Now, the thing is that while most pollen cones in conifers are simple strobili, almost all of the ovulate or female cones in conifers are compound strobili. Okay, so what does that mean? So essentially, what that means is that you have the same axis right here, just as this, but you have a leaf, and then from the leaf, instead of just one determinate strobolus, you actually have a second branch, right? Because this is just a shoot system, and we know that phyllotaxy and phytomers are arranged as such, so that you have leaf, branch, and the branch just has multiple sporangia. And we'll see how in the conifers this actually is true, and it gets it just, instead of having multiple sporangia, you generally have like one or two. But that's the fundamental difference between a simple strobolus and a compound strobolus, and this is important to distinguish. So again, like I said, the male pollen strobolus in a pine is truly no different from a strobolus in a lycophyte. It's a central stem, a central axis with leaves, and on the leaves are born microsporangia, just like in this lycophyte right here. You have a leaf and a microsporangium. Down here you have a megasporangium, so that's the one difference, right? You have micro and megasporangia on different parts of the plant in a pine, and on this Selaginella you have them on the same part. So that's the only fundamental difference. But again, you know, like we move from lycophytes and that something like this that seems so simple, but all it is just the same thing, right? It's just having, instead of producing male spores and female spores, we're just producing male spores, and those gametophytes develop within the spore wall, and then they're dispersed, right? That's what pollen is. It's a flying male gametophyte, essentially. So here's just another photo of these pollen cones. So you've probably walked past a conifer and you see these little sort of brown, like when they're old and shriveled up, they're just like these brown dangly parts. But this is the strobolus, right? Each of these is a sporophyll, right? A leaf that bears the sporangia, and on top of each of these, or on the bottom of each of these little modified leaves, is a sporangium, microsporangium. And these are in the conifers, or in the pines in particular, you have this really weird saccate shape. It almost looks like Mickey Mouse, right? And I think for a long time, people thought that these like bulbous structures were good for like allowing the pollen to fly away and like then eventually get into fly away and make its way into a pine cone. But actually what people have found is that they help, I actually have no idea like what is being shown here, but essentially the idea is that those two bulbous structures actually allow the pollen to sink into the liquid droplet that's produced on the ovule. So it's not to help it fly away, but it's to help it sink and get down to the micropyle of the ovule. Okay, so now we're back here. So now I want to talk. We talked about male or pollen cones. Now we're going to talk about these compound cones. Okay, so let me just go back here for a second. I just said that a compound cone is essentially, imagine taking this and just putting it in the axis, right? You have a leaf, you have a branch, and then from the branch, you have other branches that produce sporangia. Now, if we look at a pine cone, we can see just that, right? So in a female ovulate cone, we see that you have a main axis. And then what's this little thing right here? There's one there, there's one there, there's one there, there's one there. So these these little things right down here, that's actually the leaf. That's a highly modified leaf right there. And then this whole structure up here, this is a branch. And then on that branch, you have a mega sporangium that'll produce the megagametophyte, right? That's the ovule. So this structure, and really all pine cones, is just this, but except for, just eliminate some of these lateral branches, right? So you have axis, leaf, branch, and then sporangia on the branch. Does that make sense? It's kind of a complex, like when I first learned this, I was like, what? That's crazy. Because, intuitively you'd say, oh yeah, this is the main axis and this is a leaf. But we know the phytomer structure. We know that this is a leaf, so this has to be a branch. And here's another example of it. Again, the leaf is highly, highly reduced to this modified little bract thing. But that's the leaf, that's the branch. That's the leaf, that's the branch. That's the leaf, that's the branch. And right here, what was here before, this is obviously further along in development, but this was the megasporangium. And within the megasporangium, we produce megaspores. And in all conifers and in all seed plants, you produce you produce 4 spores through meiosis, and 3 of them die, and you're left with 1, and that's what develops into the ovule. And then the gametophyte will develop right within the spore, because it's heterosporous and it has endosporic gametophytic development. And then fertilization will happen all on the mother plant. So again, here's just a smaller view of this. You can see that here's the gametophyte, right? You can see all those massive cells in there. That's all just gametophytic tissue. And that right there, I'm pretty sure, is the archegonium, just like in ferns, right? We essentially just take a fern gametophyte, squish it up, put it inside of the spore, and put it inside of a sporangium, and then wrap it in parental sporophytic tissue, and then you have a seed. It's really all it is. And then, right, the pollen will sort of float down with those bulbous sacs into here, and then the pollen tube will germinate through here, and the sperm will be deposited into the right to the egg. It's a great system, actually. Yeah. So right. So this little leaf-like thing right here, I know it should be pointing to this little thing, but so in most conifers, you don't see the little bract because it's so small. But in Pseudotsuga, this one species, the bracts are highly modified, and they actually look like little like mice that have gotten stuck underneath. And that's actually a really good like identifying feature for this species. But essentially, it's this modified leaf. All right. Okay, and so for the second activity, you're essentially just going to be looking at all the stuff that I just went through. But one thing to keep in mind is that conifers are really, really slow at, like their whole fertilization process is super slow. The time between pollination and fertilization can be like years, like literally years. So at this stage, it's really young, it's squishy, it's ready to receive pollen. So it receives the pollen, and then a whole year can pass, and then it turns into like this state where it's extremely closed up, and like who knows what it's doing. People know what it's doing. People have sectioned these and stuff and figured out what's going on. But the pollen tubes are probably starting to germinate, and things are sort of starting to happen. And then down here, the seeds are all mature, and they're ready to be dispersed. And this whole process can take a year or 2 years, and it's quite amazing that is even possible. Yeah, and conifers are super cool, right? Like here's all this crazy diversity in the way that conifers can make their cones, right? You've seen taxus, these little fleshy berries, putting quotes around that because they're not berries, they are conifers, and berries are carpels, so they do not make berries, they are fleshy cones. So you can have fleshy cones, you can have massive hard cones that will, like if this fell off, probably kill you. And you can have really just anywhere in between. And it's quite, the diversity of ovulate cones, which are these, is actually, it's beautiful. And there is actually a diversity of the pollen producing cones too, but they're generally much smaller. And they're not as impressive. They're not as impressive as the ovulate cones. So that's all I have for you. And then the last thing I want you all to do, which we've done many times in many different ways, is to draw the seed plant life cycle. And I'm not going to do that here because I don't have like a stylist or a pen or anything, but I want you all to do it independently to the best of your ability, and try not to look it up. Just like do it from memory. Just because, yeah, it'd be good to do it. And yeah, essentially, so for the three activities that we have here, like I said, the first is sort of to examine the phytomer, and then to use a dichotomous key to key out your specimen. The second is to look at and draw the microsporangiate strobilus, the microsporangia, the megasporangiate strobilus, and the ovules, and the archegonium, and the gametophyte. And then the last thing, the activity 3, is just to, again, review the seed plant life cycle and draw the life cycle. And then when you're done with everything, take a photo and email it to me. And then that's pretty much it. Does anyone have any questions about conifers or the activities or anything?
I had a quick question,
Mm-hmm.
and maybe this has already been covered. I haven't watched the lecture videos for today or Tuesday yet,
Mm-hmm.
but like what about the structure of a lot of pine trees?
Mm-hmm.
Like essentially in a lot of like angiosperm trees, it seems like you have like a main stem, and then once you get to the top, it just kind of goes everywhere and branches. Whereas like a pine tree is very straight and almost makes more of like a triangular shape.
Yeah.
Like is that related to their differences, and like how did that evolve? It's just essentially like what explains that morphology.
Yeah, that's a good question. So I'm not 100% sure why you have that sort of like pine tree morphology. I definitely remember like reading something about it once, and now I can't remember. It is known, but I think it might have something to do with like snow. So in a lot like like that, you get that morphology in a lot of like temperate regions, and it could be like something with snow load and stuff like that. But I'm not 100% sure. I mean, you do get a lot of conifers that sort of grow weird and like gnarled in a way, more like an angiosperm, but a lot of the times that's due to damage of the leader. But yeah, I'm not entirely sure. That might be a question. You know what I'll do? I'll look into that and I'll send an email to our lab group.
Okay, cool.
But yeah. Cool. I will stop sharing my screen now. I'm back. All right. So yeah, you have until the end of the semester to turn this lab in, and we will have one more lab, I believe, next week. And what we're going to do with that one is we're going to sort of squish the flowers and the fruit lab together, and essentially I'll do the same thing. I'll give a recorded lecture, just like we do every other lab, and then it'll all be online, and you can just draw what you need to draw. and send me your notebooks. So I have all of your notebooks actually. But yeah, just draw it on, you can do it on whatever medium you want. You can draw it on a piece of paper. You can do it online. Whatever is easiest and most convenient for you all. We're going to be very lenient with everything here. So great. Well, if there aren't any more questions, then we can depart. And I wish you all the best. And if you have any questions about anything, please feel free to email me. We can Zoom or whatever. But yeah. All right.
Thank you.
See you all later.
Bye.
Bye. Thank you.
Bye.